Thank you so much, Anne and Nancy, for the introductions. And I can't live up to that introduction, but I'm going to try to talk about not 30 years in exhaustive detail, but three examples over 30 years of work that I've done that has really, I think, some tangible conservation implications or consequences. So I'm going to preface my talk by saying I think of myself as a field biologist, and I do, in fact, really like carrying heavy things up hillsides. And so this is a picture of doing field work in the Wrangell Mountains of Alaska, and this is in Kenya. But I'm not going to talk about any of the projects I do as a field biologist. And so this is a sketch that I sort of trying to summarize how I have done and worked on many different systems. And so there's a lot of nonlinearity involved. But I did start here with fireweed up at the top. Oops, sorry. And I'm going to talk about 3 of these other studies here. And they're not studies where I am a field biologist collecting data. They're studies where I'm a collaborator with field biologists, with sometimes genomicists, with other types of scientists, trying to understand species that have a conservation need, or in one of the examples I'll talk about, a sort of general understanding of how rare populations work. And so I'll show you at least a good smattering of pictures of cute animals in pretty places, but these are not places where I collected the field data, and I just for fairness want to make that clear. So I've had the opportunity, so here Nancy did a great job, except she really stumbled over purple gorgonian corals from the Mediterranean. And so I've worked on a whole variety of plants and animals in this context of basically being a population modeler, and especially a population modeler trying to understand conservation strategies and approaches for rare species, of which all of these are. So what I'm going to do today is talk about 3 of these species. One, and I'm going to talk about some subset of the work I and my collaborators have done looking at them. One is the Channel Island fox, which I'll start off with. One is, and they live in California, as does the last one. Oh, I said, oh, sorry, the numbering was off. As is the last one, the California condor, which actually lives closer to us, like in the Grand Canyon, and is native to Colorado, although extirpated here. But because these two were not California or Colorado natives, I'm going to talk a little bit about Saussure weberi, a highly endangered or highly rare species, I really should say, of the high alpine that is native to a very small set of areas, including in Colorado. And I'm going to try to talk about these to give examples of where we have successes, and especially in the case of the California condor, where we still have some really serious remaining challenges to doing conservation. So I'll try to show how careful scientific analysis and collaboration can actually have really tangible benefits for species conservation. And I'll tell you part of my subtext of this part of this goal is to try to convince you that it's not just politics that causes conservation to be difficult. It actually is science too. There's lots of things we don't understand or require more careful understanding of to basically do effective conservation. A lot of people I know, like undergraduates in a conservation biology class, say, well, really, it's just a matter of politics and like policy failures. It's nothing, we don't need to do science anymore. And I would argue that's not true. I'll discuss how modeling not only the species involved, or sort of understanding and analyzing them, but also the management strategies and even the sort of people doing the work can actually be as beneficial in understanding rare species problems. And then I'll try to show how we're still gaining ecological understanding very specifically about how rare populations work. And basically, there's difficulties of understanding and appreciating ways that rare species, including ones we've made rare or naturally rare species, actually persist, which are really important to understand to basically go farther in furthering conservation goals. I'm going to take a moment to say 2 things. So the first is, a lot of people question, like, why put all of this effort into rare species at all? And I'd say there's 3 reasons, for me at least, and for a lot of people who work on rare species. One is ethics. There's a very tiny number of critically endangered or threatened species in the world right now that are not critically endangered and threatened because of human activity. And so ethically, even though species have gone extinct over the millennia naturally, we've deliberately or undeliberately caused most of the extinction risk and extinctions in the last several hundred years and going into the future. And I feel there's an ethical reason as a scientist or just as a regular person, a normal person, to try to prevent. One is ecology. Some of those rare species are things like sea otters, which are critically important for entire ecosystems. And the more we understand about how ecosystems work, the more we find that species that are naturally rare even sometimes play critical roles in structuring communities. So if you care about ecological functioning of the wider world, rare species are also worth preserving. A sort of subtext of that was something that mentioned in the blurb for this talk, which is that most species are rare. This is just sort of a natural, I don't know, what would you call it, Christy, a law. Like that basically there's a really skewed distribution of abundance. So like you go to a redwood forest in Colorado or a ponderosa pine forest in California or Colorado, most of the individuals you see are the species that habitat is named after. The vast majority of species are unusual and rare. And so rarity is common. So if we give up on rare species, we're giving up on most species. And finally, this last point is basically it's really easy to say, at least in theory, it's easier to say what is a rare or endangered species. It's really hard to say what's an endangered habitat or ecosystem. And so laws to preserve biodiversity are basically focused on species for a very pragmatic reason, and they therefore are the tool by which you try to save broader landscapes or restore them. I'm going to say one other thing on this slide. Just before we got ready to give the talk, my children, who are here in the audience, told me everybody hates Comic Sans font. And so I'm going to apologize to almost all of you who are haters. But I think maybe because I give talks a lot that meld modeling results, simulation results, and trying to make it seem friendly, I like Comic Sans font. So my apologies. I'm going to, I apologize to my children in particular. So the first species I'm going to talk about is the island fox. So I've been involved working on the island fox for about 25 years. So this is a classic island endemic species. It only lives on islands. I say it's classic in multiple ways. It's related to the mainland gray fox, but it's much smaller. It's the size of a sort of house cat. They're very tame to humans and other potential predators. And they only live, they only live on the Channel Islands off of Southern California. I'll talk mostly about the populations on these 3 islands in the northern Channel Islands, although they also live on San Nicolas and Santa Catalina Islands. So there are 6 subspecies, one for each island they occur on. 4 of them were listed under the Endangered Species Act. And I say were because they've now been delisted, and that's part of my story. This work, and I'm going to try, I don't actually have a really exhaustive acknowledgement slide at the end, but I'm going to try to acknowledge some of my key collaborators as I go along. So in this work, Vicki Bacher started off as a postdoc with me on this work. She's now a research professor at Montana State University. And Scott Morrison was and still is the head ecologist for the Nature Conservancy of California, the largest state branch or any branch of the Nature Conservancy. And he was really instrumental in the work I'm going to talk about, providing resources and sort of leadership and innovative thinking. So why were foxes endangered? So on the northern islands, an entire slew of introduced ungulates were left and to basically run wild on the northern islands. So, I keep doing that, sorry. So sheep, pigs, cattle, mule deer, actually goats aren't listed there, but they were introduced, and even Roosevelt elk were introduced to those islands, which had no native large mammals. The fox was actually the largest native mammal, about this big. They caused extensive erosion, as you can see here. And they also favored widespread invasion of invasive plants that basically resulted in habitats that basically have very little food for foxes or lots of other native species there, including a lot of the endemic birds. So like this is a stand of introduced European fennel, which is basically a monoculture provides very little food for things like foxes. But none of those are as important as the direct effect of golden eagles, which I'll talk about on the next couple slides. On one of the southern islands where there was endangerment, Santa Catalina, actually it was a different problem. This has a large amount of private land, and basically people have continued to keep bringing dogs and cats and even things like raccoons that carry canine distemper. And there have been a couple different outbreaks that have wiped out to very low levels the fox population there. So on the northern Channel Islands, the problem was this noble creature at the bottom, a golden eagle, and the history of how they became a problem. They're a native species in North America. They're not an introduced species, but they were not present on the Channel Islands, basically historically. That's almost certainly because there were abundant populations of bald eagles there, which basically feed almost entirely on birds and marine resources like fish. But they basically are dominant territorially to golden eagles. And so they basically, because there were large populations of bald eagles, there were no golden eagles. During the 1960s, actually when I'm showing you up there on the picture of this map, at this point in Los Angeles was the largest chemical plant in the world making DDT. And they dumped the DDT effluent and byproducts into the ocean. And so to this day, there's very high DDT, DDE levels in the waters of the Santa Barbara Bight, which is this body of water surrounding all the islands. And so those DDT levels cause the extirpation of bald eagles there. That led to an opening with a considerable delay where golden eagles, as they began to increase after persecution on the mainland, began to go out and find there were these islands there that they could also use, and quite rugged islands with good sites for areas for the nests of the eagles. There were also abundant introduced prey, especially feral pigs. So a full-size pig is not prey for a golden eagle, but especially in the mild climate of Southern California, feral pigs, introduced pigs from Europe, basically reproduce year-round, producing little juicy piglets. And so juicy little piglets are great prey for golden eagles, and especially in a habitat which doesn't have many other, especially sort of 12-month of the year prey items that are abundant. And so that led to an increase over the 1990s of golden eagles. And so through the 60s and 70s and the 80s, there was this slow beginning increase in sightings of golden eagles. And then in the 90s, they just shot upwards. Coupled with that, foxes, these two different lines are fox populations on two of the islands where monitoring was ongoing during the 90s, crashed by about 90 to 95% over less than a decade. And so you'll notice this slice of time is just a little slice of time here. I'm not going to talk about skunks. You can ask me about the skunks. There's also an endemic subspecies of spotted skunk on the islands. And I'll tell you about those if you really want to know later on, but I'm not going to talk about it now. So by 2000, populations of foxes on all these islands had crashed to the point where the land managers, mostly the Nature Conservancy and the Park Service, actually made their best effort to actually trap and bring into captivity all the remaining foxes. They didn't actually understand initially what was happening to them, but they knew that they were declining rapidly. By capturing foxes, they found out it wasn't an epidemic disease, which was the initial thought. But basically, the situation got so dire that the largest number of foxes on any of the islands was guessed at around 30, where there had been many hundreds before. And so they brought them all into captivity. And as that process was going on, there began to be this hypothesis that basically it was the golden eagles, which had shot upwards in numbers, that was causing this problem, basically this basically incredible predation rate, driving fox populations down. That seemed sort of unlikely. There were still not very many, I'll try not to use the pointer as much as I can, not very many eagles relative to the number of foxes that existed there. But some of the eagle biologists who were brought in basically sort of pointed out, well, eagles can eat baby pigs, but except for really, really young baby pigs, they're too heavy to be carried in flight. And when you're feeding nestlings, you need prey which are as big and juicy as possible, but what you can carry many miles to basically an eyrie to basically feed to your chicks. And as the decline was going on, the fox biologists began to put radio collars on the foxes. That hadn't been common practice before. And so this is showing one very guilty-looking eaglet surrounded by dead foxes with radio collars. And actually, this became, this is just before basically all of the foxes that they could catch were caught. Basically, this became the way you found the Eries. Basically, you follow the radio collar signal and basically found these piles of dead foxes around these chicks. And so that made it seem pretty likely that this was the problem. And so that led to substantial efforts to do a whole bunch of different things. One was to capture and translocate the eagles. So no golden eagles were killed, but they were basically captured and taken. Where would you take an eagle you didn't want to have come back? Nevada. So they were mostly taken to Nevada, where there was a much slower recovery of golden eagle populations following DDT and shooting persecution. And as at the last I've read or heard from any of the people involved in that, none have come back, which is sort of interesting. You'd think that they might have like beelined it back, but that has not happened. There was also a recognition that if you're going to get rid of the eagles, they're subsidized by the feral introduced pigs. So there was a complete eradication of the pigs and all of the other ungulates, which had mostly been eradicated by that point. And then there was all of the foxes that had been captured were actually in cages, captive breeding program to try to boost their population, not on the mainland, but on the island. There was a real fear of bringing them to the mainland where they might get diseases and never be able to be brought back again. And there was even more, and the even more that I'll just mention briefly is there was also an effort to reintroduce, by hacking out chicks of bald eagles to basically reestablish a bald eagle population. That actually has been fairly successful, and that's probably one reason that the Goldens haven't come back. Actually, there now is a population of bald eagles. So while all of this was going on, I and Vicki and Scott and others were asked to basically try to address some of the management questions involved with how bad is it, how quickly could the population recover, how many eagles could you tolerate eating foxes and still have a fox population that wasn't crashing, that was basically stable. And also some other questions like, when should we release the captive foxes? Like, at what point will it be safe enough to basically release them? And so I'm going to talk to you mostly with a picture or two about our effort to build models for foxes and then models for the people managing the foxes to try to answer some of these questions. So one of the sort of basic tools that I'll bring up for foxes as well as for condors later in the talk is a simple demographic model. So in this case, we have young foxes, ones who were born this year, and we basically have adults, including young adults. A fox can survive or die. If it survives, it can also, with a certain probability, breed, and you have a certain litter, and you have a certain statistical distribution of litter sizes. And if you can estimate those numbers, which we could from past research for the foxes, you can basically make a model for how the whole fox population works as a function of survival rates, reproductive rates, and basically that drives a population model. And so this is exactly what the Census Bureau does with human population data to estimate like how many kids will be in our schools in a decade. But we're doing it for foxes who never go to school. And so those survival rates and reproductive rates, though, it's more complicated than I'm showing here because they depend on fox numbers. Foxes actually compete for food when their numbers get high, and they actually have problems finding mates when their numbers get low. The climate matters a lot, especially El Nino events. Eagle numbers, obviously, that basically reduces survival rates. And also, there's pretty strong habitat differences across the islands. And so this is sort of a little bit of an aside. What do you do with a model that says how many foxes will there be with some random chance there'll be an El Nino event or La Nina event next year or a certain population size? And a lot of the game in conservation is to basically say, well, is the population viable? Is it basically likely to go extinct under some certain amount of time with other certain sort of criteria? And the answer to that depends upon what a model like this gives you, which is population growth rates and how variable they are. But it also depends upon some things that are actually human constructs, like how far in the future, do you want to estimate whether a population is viable or not? The most extreme difference of opinion about this that I've ever been involved with was for Yellowstone grizzly bears, where on one of the initial sort of arguments of is the population viable, environmentalists said we should look over basically viability over something, actually, sorry, I reversed that. The government, the US Fish and Wildlife Service, wanted to analyze viability over 20 years. The environmentalists involved analyze it over 1,000 years. And you get a very different answer about the risk of extinction if you're talking about a millennia or about the lifetime of a single bear. So there's no right answer there, but that's basically a discussion that you have to have about your values. How safe do you want things to be? The other aspect is whether the population is really good size, or in this case, like not very good size. Like you could start off with a population slowly declining from 10,000 animals, and it's not going to get to some critically low number anywhere near as fast as one that starts at around 50. And finally, and I'll use this term, so keep it in your mind later in the talk, there's what we would call quasi-extinction. So it's very hard to predict when the last passenger pigeon, Martha, went extinct in the Cincinnati Zoo. It's much easier to say, when has the population reached such a low level that problems like inbreeding become critically endangering, things which are sort of added problems beyond sort of the problems that any population faces. And so you often try to analyze the quasi-extinction probability, not the true utter extinction probability. And so with those numbers, I'll talk a little bit for the foxes about how they were agreed upon, you can then run out a simulation model. And this is the other sort of part of this way of approaching, is a population endangered or not, or can we do something to make it significantly less endangered? To predict the future, what you really do is try to run out many, like this is actually showing 100, but in reality you do about 10,000 of these, random futures, and ask how many of them drop down to a critically low number below the quasi-extinction threshold. And that number out of all the random futures turns into the probability that you use to try to guess at what's the probability of the single future which will really play out that will actually see this sort of catastrophic result. And so and I'll just sort of show you like how that works with eagles. So here's probabilities for one of those islands, and all the parameters of running out 100 futures, assuming there were only 4 eagles present every year, 5 eagles, 6 eagles, or 7 eagles. And so with 4 eagles present, and you start with a fairly high number, basically there's a pretty low probability, about 4%, that over 100 years the population would go extinct. If you go to 6 eagles, still a pretty small number of eagles, that adds up to about 53% probability of dropping to this very low number. And even one more eagle gets you to 92% probability that you would guess that you're going to basically see catastrophe for the foxes. And so with this kind of an approach, you can then say, well, how endangered are the foxes with a certain number of eagles? And basically, how would we know? This is one of the questions we initially tried to answer. Like, if you were here taking data on, say, the survival rates and the population sizes of foxes, how would you use that data to guess that you were in this situation and not that situation? And this is part of the goal of trying to ask, how should you, in this case for the foxes, how should you monitor the foxes? And so this is one of the very first questions we tried to answer. So we used all the existing data to basically make that model of foxes work as realistically as possible. And then we asked, like, how do you monitor foxes? And there were 2 ways that was done, and still is done. One is basically by trapping them on trapping grids to basically get population size and actually try to recatch the same individuals to say something about survival. But the other is to put on these radio collars which have what's called a mortality signal. When an animal dies and stops moving, basically the collar makes a different signal, and you can actually, without ever finding the dead fox, know that it's dead. And so basically the collars and the grid trapping are the 2 ways you monitor the population. And we found that if you use both of those kinds of data, and you say, what's the adult population size, and what's the annual adult mortality rate? Basically, there's much more variability for the young foxes, so basically they just sort of add noise. you can basically make a pretty good prediction of the quasi-extinction threshold, the threshold of dropping below that critically low number over the next 50 years. We actually also found that if you average the data over 3 years, so you basically take those two different metrics with a 3-year average, you get something which is what you really want for this. You get something where you basically don't have, it looks like Boulder Creek up there, you have something which is like Niagara Falls. Basically, either there's basically no probability of extinction, they're perfectly safe, or with a very, very narrow gray zone, they're critically endangered. So you basically can make a pretty clean, scientifically based, data-based estimate of basically risk of extinction. This was the first thing that we did, which the managers really did not like. Because, as you might guess, it's really expensive on rugged islands like the background here to put out traps twice a year, hundreds of traps for weeks at a time. It's also very expensive to trap foxes and put expensive radio collars on them and have someone monitoring them. And they really wanted us to tell which of these two things is the best. And there's lots and lots of results we found that showed that neither one worked very well. You needed to do both of them. And so our answer was basically you have to do more and more expensive. But it was based upon really trying to analyze the situation to get a meaningful signal from the data that could be collected. The other thing that we worked on initially was how do you define what a recovered population is? And so how would you say that a population, based upon the data you're collecting, as it hopefully at that point was going to be recovering, will basically tell you that this population is basically safe now, or at least reasonably safe. And so there are sort of a few different ways to think about this. And I'll say at the onset, let me see. Yeah, I still have Nancy's pointer is still hanging in there. So under the Endangered Species Act, the ESA, you have to define what recovery is. Basically, you say something's endangered or threatened, and then you say, well, you also must define what will tell us that it's recovered. And mostly that's done by something like 100 animals. Like 100 is good enough. And actually there's a lot of analyses of recovery plans that have shown that the magic answer is usually 3 times as many as you have when they were declared endangered or threatened. And actually, Doug Shemsky did that work, a friend of ours. And basically that's not very scientific, it's just very convenient. And so we wanted to do something better than that, to try to say, well, what is a really good measure of the safety of the population, at which point we'll know that these populations that were so endangered they had to be captured in mass, well, not really in mass, there weren't that many of them, basically we're at safety again. And so this goes back to that diagram, though, of defining population viability. There are some really arbitrary, or at least human, sort of ethics and policy decisions that have to be made to then start to turn the scientific crank to answer. And we found it was very hard to get the managers, especially from the Park Service, to decide how safe from extinction recovery would be, because they had just watched this species, which is quite charismatic, almost decline to complete extinction. And so they were not comfortable basically being in the position of saying, like, there's some probability of extinction, but that's safe enough. So we did what you always do when you want to get someone to do something hard. We took them to Davis, California, in the summer, and we went to a room at the university that doesn't have air conditioning. And we served them really bad coffee out of styrofoam cups. And so we convened this workshop for several days in Davis under those circumstances to basically get the managers to tell us, well, what is safe enough? And this is what they came up with, which is actually, I think, it's something which is comforting to me, and many people who work in the environmental field who don't work for the government sort of think of the government biologists as like bureaucrats. And I actually think that most of them are really trying hard to do the right thing. So they argued that the current observations that we would have should indicate, it's now off, less than 5% risk of falling below a quasi-extinction threshold of about 30 foxes, that's what we had just survived, for 50 years, which is many fox generations. They're fairly short-lived. And so with that number, we could then make graphs like this using the simulation models that I've shown you. We'd say, well, adult mortality rate and adult population size, these 3-year averages, will give us a way to estimate the risk of extinction. So that this critical less than 5% is this isocline for risk. And if we have a smaller population or higher adult population mortality, you get up to here things like 70% risk. Oops. Oh no. Okay, here we go. Sorry. But the 5% risk isocline is really the one that's most important here. And this is showing that this was different for different islands. For Santa Cruz Island, which is a much larger island than San Miguel, you have to get to a much larger population size to be at the 5% risk isocline. And the managers further said, and also, you basically have to be down in this zone for at least 5 years in a row, and there's uncertainty with any real data. So the isocline, basically that estimate here, bounded by its uncertainty, all has to be for 5 years down in this zone to declare recovery. And that's actually a really, really rigorous criteria. And one reason I think that the Fox recovery was as successful as it was is that this is a rigorous enough criteria. There was no argument that basically this was not good enough. The science wasn't good. Like, this is a hard criteria to meet. And basically, if you meet this, everyone agreed you've recovered the foxes. So I'm sorry this is so fuzzy. This is the only version I have of the San Miguel extinction isoclines with the data. So again, here's the 5%. And basically, once the recovery plan was put into place with these criteria, this is what it showed year after year. So basically, quickly you got out to here, and basically you'd met the recovery criteria. Basically, every year for more than 5 years in a row, you were down here in this zone of fairly high safety. That wasn't true for all the islands. So this is showing for Santa Rosa. Basically, for many years, we were up here sort of trending gradually in the right direction, but not meeting that criteria. And actually, this was the last of the islands to basically be declared recovered. And then finally, you began to get to these much better numbers, mostly by having much lower mortality rates of adults. In 2016, that actually led to the recovery of all of the populations that were, I should say, the northern islands where golden eagles had been the problem. The Santa Catalina Island population was basically downlisted to only threatened. The other ones were declared completely recovered. And this was pretty amazing when you look, so this is showing for San Miguel Island. These are the estimated number of foxes before golden eagles, during golden eagles, and afterwards. And this was the fastest recovery of any endangered listed mammal ever under the Endangered Species Act. And so partly it's because foxes are really good breeders, so their population could bounce back rapidly, but partly it's because there was this concerted effort to solve the problem and try to understand the problem. So going back in time, I just want to mention one other aspect of this study. One of the things which was most problematic when we were initially involved, and basically when the initial efforts to basically reverse these declines and get rid of the threat, especially of golden eagles, was how to capture eagles, or whether it was possible to capture eagles effectively enough to actually cause them not to be basically an overwhelming threat for foxes. And so eagle predation has to be kept to very low levels, as I showed you with one of those spaghetti graphs of many, many iterations of the future. And that was a problem because capturing eagles is really expensive. At the time that we did these analyses, the estimate was it took $40,000 to capture an eagle. And there were dozens of eagles, and more coming all the time onto the island. It's also just extremely ineffective and slow. And so this is just showing an analysis the data at that point. So the number of human hours of efforts, basically these are employees employed to stay out on the islands looking for eagles and trying to capture them, had ranged from basically less than 100 hours per year up to basically around 700 hours per year. That was the range that actually been done. And even at the high end here of that real range, the chance of basically capturing an eagle you were trying to capture within a single month was less than 5%. And in the meantime, that eagle is eating foxes. And so we basically tried to answer the question of, like, can you ever do it? Can you ever actually capture eagles well enough to basically control them as a threat if we ever, at that point, if we ever actually start to reintroduce foxes? By building a model that's basically not just for eagles and foxes. So we have here foxes, this is the demography of foxes, reproduction and growth, being eaten by eagles. And then we also basically simulated the process of sampling with grid trapping and collars, the foxes. And we then also simulated the decision makers, like the bureaucrats, trying to say, okay, now that looks bad now. We should basically, we see a signal of lots of more mortality. We should start trying to capture eagles. And when their mortality goes down, we can stop because it's really expensive. And then in the meantime, in the interim, we're trying to remove eagles. And we tried to do this for a huge range of sort of circumstances, like weather conditions, and basically different rules for, say, stopping and starting. I don't want you, I couldn't figure out how to make this all fuzzy, so you couldn't actually try to read it. The only thing I want you to get from all of these complicated graphs is that there's very little dark blue. So what these are showing is different combinations of all of those parameters, including vastly more effort than they'd ever put into capturing eagles. And only with vastly, vastly more effort do you get to these small areas of blue over here, which would tell you that basically it's good enough so that the eagles can recover. So this was the second thing that we did, which was really unpopular. Because we basically said, you're doing a great job, except it'll never work. This is never going to really solve the problem. But the upshot of this, and this is where Scott Morrison, I think in particular, really had this sort of, he sort of understood what we had shown him and said, well, we've got to then just figure out some totally different way of capturing eagles. And the answer was that it was cheaper and far more effective to hire these sort of like cowboy tiny helicopter pilots from New Zealand to fly these very small agile helicopters over the island and basically fly after eagles when you found them, tiring them out until they would land and you could drop a net on them and capture them. And if you've ever hired a helicopter, you know, helicopter time is very expensive, Bob Anderson. And yet, this was so much cheaper than what they had been doing that this became the way all the rest of the eagles were captured. And so our contribution as modelers was basically just to say, what you're doing really sucks, and you've got to do something different. But without that analysis, I think they would have kept on doing this kind of lame, not very effective management method. So I want to quickly say, up to the present, we're still working, me and Vicky and others, on various things on the Channel Islands and on foxes. Well, some of the other things we've addressed is different strategies for vaccination, for rabies and distemper for the island foxes. Also, this is an endemic bird, the island jay, which is very susceptible, we suspect, to West Nile virus, which isn't present on the island, so trying to understand how to cope with West Nile virus becoming more common with climate change. And then also, we're continuing to work on one of the Navy-owned islands in the south, San Nicolas Islands, which is this highly degraded, very uniform kind of habitat where the foxes have very different dynamics and require different kind of management methods. So I want to turn now, very quickly, I'm sorry, I get so enthusiastic about foxes, to talk about this native plant of Colorado. So the dwarf sawwort, or I'll call it Sausuria, because that's what I know, that's what I think of it as, is this alpine rare species. And that actually is a rare thing in and of itself. There are not very many rare species in the alpine. So this is what it looks like. It's a member of the daisy or sunflower family. It's naturally rare, so it's not really endangered in the sense that something like the foxes were. Basically, it naturally occurs in this handful of habitats up to Montana and down basically to the Mosquito Range near Leadville. Everywhere it's ever been found, it's very uncommon. It basically forms small, sparse populations. So this was a data set collected by Ron Abbott, who I'll talk about in a second. I don't have a picture of Ron. But it was analyzed by my then postdoc, Megan DeMarsh, and I. And this is a picture of Megan here. She's now a professor at University of Georgia and with two of our undergrad assistants. So from 1996 until the present, like last year, Ron goes out every year and basically counts the number of these plants in Horseshoe Cirque, this is a picture of Horseshoe Cirque near Leadville. And every year he does this, not because he's not a biologist. He actually has a master's degree in pollination biology, but he worked in a hardware store for his whole career. He just liked doing this, and he still likes doing it. And basically in 49, there were 50 initially, as you might imagine, 1 meter square plots. He counts the number of these plants every single year. And he continues to do that, and we actually would like to now revisit the data set again. But this data set is unique and let us do something and sort of look at a common assumption in all the analyses of rare species, which is that except for something, if you're a plant biologist, something like the size of the plant, or if you're something like a bird biologist, the age of the bird, something very obvious, all individuals are the same. Like if it's a wet year, everyone might do better. If it's a dry year, everyone will do worse. And that's an implicit assumption of most analyses of rare species. And we were really curious using Ron's data, where he has these individual plots, many of them, that every year he censuses separately, whether that was a good assumption or not. So this is the data for all the plots together. And this is the 20-year data set we've analyzed. So here's the total numbers. But we were curious whether basically these fluctuations in the total population size sort of is concealing or is being driven or influenced in some way by individual variability. And you'll notice this looks like the population is declining to extinction, but notice this is 1,300 and this is 1,500. So basically these are all a relatively small bounded set of numbers. So there's two ways that we are particularly interested in looking for this individual effect. One was asking whether in some of these 49 plots, so here's time and some measure of like population size or growth rate. You might imagine that in some of them, there's huge fluctuations, where there might be good years, but also really, really bad years, whereas other ones might be more buffered from that variation. The other possibility is that there's actually sort of reverse trends. So in some years, maybe this population does well, another population does poorly, and then vice versa under some other condition. And so we looked at Ron's data to try to understand whether that was really going on. And this is potentially a way to understand how does an inherently rare population, a tiny rare population, survive at all. So here's what the data look like. These are actually data, not made up data, like the other squiggly lines I've shown you. So here's for those 49 plots, and I just did 2 different colors to make it easier to tell the different lines, showing fluctuations in numbers. And the only message from this is there's a lot of variability. They're not all going up and down in synchrony. This is population size, but if we look at population growth rate, it actually is easier to read. So 0 means a population that is basically stable. And you can see, and now the colors are not random. So basically I said every population that declined in 2005, I'm going to make black, and the ones that increased in that year, I'm just going to make red. And it allows you to see those black populations often track together. They basically seem like they're correlated. And some years, a lot of the red populations also do the same thing, but in many years they don't. There's basically different kinds of uncorrelated or even negatively correlated patterns in basically the population growth rates. Of these, all very close together, like the size of this room, all of these plots would fit in little subpopulations. You can also just look at the correlations between all the different pairs of those plots in the growth rates. And you can see a lot of them are positively correlated, that's the blue, but there's also quite a lot of negative correlation. And so that would imply that there is a lot of this individual variability. We also, the next thing and the last thing we wanted to do with these data was to basically ask, does that matter for how the population works? Basically, the individuals, little subpopulations do different things in different years. So we simulated over 100 years the population growth rate, either with the observed sort of messy mix of correlations in how different little plots performed in different years, or assuming the same amount of variability but perfectly synchronized. And then we asked, basically, what are the chances of seeing a population decline by a certain amount? And so that's what this is showing. And so here's what you can think of as the quasi-extinction threshold. I told you it was going to come back. Basically, by how much, what fraction of the initial population would we define as the extinction threshold? And then here's the probability of decline. And I'll explain this graph a little bit more in the next slide. But what you can really see here is if you assume synchronous variation, it's a really different line than if you use the real mix of highly synchronized for some pairs of plots, but often very unsynchronized variation from year to year. And the easiest way to see this is to think about what's the probability of declining by 50% of your initial size. So that's here. And for the real population with a real mix of correlations, the probability of declining that much in 100 years is 0. And if you had everyone actually responding to the environment in the same way, the probability is about 50%. Probability declining by that amount. And you see, if you go down to like declining to even 20%, a much lower risk, there's a much lower population size, there's still a significant difference in the risk. And so this is essentially showing, and this idea in economics are called portfolio effects. Basically that unsynchronized, this is why you have a diversified stock portfolio for your retirement account. And actually this is a term that ecologists have borrowed from economists. To basically, these portfolio effects seem to actually really strongly stabilize this population, another population of another rare plant in Wyoming we've analyzed, and people have shown the same thing for like salmon runs, also at much larger spatial scales. This is something where I feel like we're only beginning to understand that this is a critical part of having natural populations maintaining themselves more or less stably in highly variable environments. And actually, this work has led to more and more analyses of this same question about how and when will climate change basically overwhelm the sort of buffering ability of populations, partly through this mechanism, to persist. So a little bit of climate change, often you see no effect on populations, and then at some point it basically begins to break down. And we suspect that these portfolio effects are one reason why. So I'll say one more thing to tie this back in. This is one reason we're so interested in the foxes on that degraded landscape on this naval island, is that it's much less variable, and we don't think the portfolio effects can buffer that population anywhere near as well as the more natural islands to the north. So I'm going to skip this because I'm going to be over time. Can I keep talking a little bit and be a little bit over time? Is that all right? Okay, I would really like to talk a little bit about condors. Wait, where's Chris? Chris, is that okay? Okay. So the last example I wanted to talk about is the one where we actually have some of the most sort of worrisome, intellectually interesting but worrisome results. So California condors have a lot of complications with recovery. They're really large vultures. They have very low adult mortality rates and also very low reproductive rates. So unlike foxes, they actually are not very resilient in the face of something you do for them, because it takes them a long time for the population to do anything. This work, this is actually from 2016, a group of us who had a workshop on how to try to analyze and understand better endangerment and recovery options. This is Vicky Bacher again, this former postdoc of mine, and here's Myra Finkelstein, another former postdoc. And they and I sort of are this sort of population modeling team that works with these field biologists on condors. So condors have been trying to go extinct for about 20,000 years. And so in the Pleistocene, they occurred in Florida, in Maine, all the way down into Mexico, as well as all the way up into British Columbia. With the extinction of the megafauna at the end of the Pleistocene, their range shrank dramatically. And so it makes sense. They're the largest vulture we have. Basically, they need lots of big dead things to eat. When the big things are no longer alive and therefore can't die, there's less food for them. By the 1880s, they were restricted to Western North America. And by the 1980s, they were restricted just to Southern California. And by 1982, there were only 22 left in the wild. So currently, at that point, basically they were all captured and brought into captivity at the San Diego Zoo breeding facility. And basically, efforts were made to basically increase their population size, initially just in captivity, and then basically by releasing them. So there are now about close to, actually, this is done last year, about 600 total. There's about 350 spread across 4 populations in the wild. And 2 of those, the 2 that actually they're sort of mixed here, but there's 2 of them in California, one in Baja California, Arizona, and the Grand Canyon. And then now there's a new release site right up here at the Oregon, California border that the Yurok tribe, for whom it's a sacred bird, has really pushed for very hard. It's a very expensive effort. So about $40 million have been spent. So far, it's about $2 million per year. Until I went to a whooping crane conference, I always said this is the most expensive endangered species recovery. But the whooping crane people said, no, 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 no. Ours costs even more. And so the Endangered Species Act recovery goal, which I had nothing to do with trying to analyze anything for that, specifies that recovery could consist of 2 self-sustaining, so you can think about viable populations, of at least 150 individuals each. The populations now, tentatively at least you could say, meet that goal, at least in California, are getting close to meeting that goal, except for the self-sustaining part. Basically, here's the cumulative releases from the captive breeding program. And here's the actually annual population size as a cumulative total. Here's the number of wild fledged, basically natural reproduction. And here, and the red line is the cumulative number of dead or removed. Removed basically means a condor that got so sick or injured had to be brought back into captivity. And so just looking at these lines, the only reason we have this growing population is because of this like outpouring of captive bred young condors that keep being released into the wild. Basically, the number of cumulative dead, is almost enough to match the total population size. And so basically, it's only by being subsidized, not being self-sustaining, that we have condors in the wild. Why are they not self-sustaining? Now I'm going to try to move fairly quickly, sorry. It's because of lead poisoning. So if you eat dead carcasses, dead mammalian carcasses exclusively, and you like large ones, large dead mammalian carcasses usually were shot by somebody. There's some of them that die for other reasons, but in the landscape of California, especially, basically this is basically things that were shot. Most bullets are still made with lead. Most birds, condors included, will ingest things that are like gravel. So if they encounter lead in the carcass, they'll accidentally or preferentially eat the lead bullet fragments, and that leads to lead poisoning. So the blood lead levels in condors, this is through a long swath of time, basically remain very high. So every condor there's an effort at least every year to capture every single condor and at least once take its blood lead levels, as well as measuring other things for basically the health of the bird. Just to put that into context, if you ask what are the data from human children who are almost only ever tested for lead because you think that they were lead poisoned by eating like lead paint, the average is 0.05% have lead levels of 450 nanograms per milliliter. 22% of condor samples have that level. And since condors are repeatedly sampled, about half of all free-flying birds have at least once had that level. And then we initially basically asked, well, if you could get rid of those lead deaths, the death and basically the recapture of birds that were killed by lead, what would that do to the population? And so that's that yellow line at the top. So this is basically going forward for 20 years. You'd say if you could magically get rid of lead poisoning, the population would be slowly growing. Condors will only ever slowly grow because they have such low reproduction. If you do what you're doing now, and that includes things like if a bird looks sick, you capture it and give it chelation therapy. So many of the condors have had chelation therapy. And then you basically nurse them back to health before you release them. You basically have a population which is completely stable. If you say birds die 100% of the time if they hit 1,000 nanograms per milliliter, that's actually much, much more than lethal for humans. The population would decline fairly rapidly if basically you stopped doing captive releases. This is all in that context. And if you say they can tolerate 3,000 nanograms per milliliter, which some condors definitely can do, the population will still be going down. So basically this is saying self-sustaining equals getting rid of lead. there's been important policy progress, partly as a result of those kinds of analyses, to try to solve this problem. So in 2008, California restricted the use of lead ammunition in the condor's range. And then subsequent to that, Jerry Brown signed another lead ammunition hunting ban that was phased in from 2013 to 2019. And so by 2019, in theory, no lead ammunition for rifles could be used in the state of California legally. And so these legal efforts have also been supported by a really intense amount of public outreach to try to get hunters to use non-lead ammunition. So the sad thing about that is there's been no evidence at all that that's been working. And so what I'm showing you here for the Southern California flock, I'll tell you what that means in a second. Basically, deviations from zero are basically deviations in the amount and frequency of lead exposure in condor blood samples relative to the period basically before 2004. So remember, 2008 and then 2009 are the first ban and the full implementation of the second ban. And what you've basically seen since the bans went into effect is growing lead contamination, not shrinking. And so what we'd like to see are these lines here basically below zero, and we're actually seeing them consistently above. And basically, I'll show you several other graphs that look like this. What's being shown here is targeted and age and release are basically controlling for these effects. So like birds which have been out in the wild longer tend to get more lead contamination because they basically are better at foraging for basically natural sources of food. And basically younger birds or much older birds also tend to get into trouble a little bit more. And so I'll show you a little bit more analysis for the Central California flock and the Southern California flock. So for this two different flocks, basically you see the same thing. So again, rising lead, not falling lead, subsequent to these big policy victories in terms of laws outlawing lead use. You can, though, account for all of that increase in the Southern California population if you account not just for age and release, but also the behavior of the birds. So the behavior of the birds is changing. And if you control for that and ask what's sort of the remaining pattern in the data, it's actually for continuously lower lead contamination. And so what's happening is the birds are acting differently, and that's driving their increased exposure to lead, not something which is sort of a failure of that general policy. So to understand what behaviors have changed, it's fairly simple. It's basically where they're feeding. So when condors are initially released, they're released around proffered feeding sites. So this is a dairy calf that died naturally that's been set out because it doesn't have any lead in it. It's basically a clean, proffered source of food. And I'll just, because this is fun, this one was set out at 2:02. Less than an hour, a couple condors came in. At 3, a lot of condors came in, and by 4:40, there was nothing left to eat. And so condors are really good at their job. But this also sort of exemplifies this series, one of the problems, which is condors are very social, and they watch each other, and they see where they're landing, and if a condor finds a food source, lots of other condors go there. And so you might think of them as like teenagers, and sort of if you hang out with the wrong crowd, you're going to get in trouble. And so one of the things that happens is the condors spend more and more time out in the wild as they watch other condors and go farther and farther from these proffered feeding sites and basically eat more and more lead-contaminated carcasses. The other thing which is unfortunately happening also as a result of that dynamic is that fewer condors are feeding on marine mammal carcasses like this whale on the coast. And so if you feed on the coast, your chances of being lead poisoned are much, much lower because you're feeding on these seal carcasses and also whale carcasses that basically are clean sources in terms of lead at least. So if you account for that, you can basically understand why this pattern looks like basically there's been no progress in basically controlling lead in the South. In the Central California population, that's actually not the case. So again, you account also for this shift in behavior, and it has almost no effect in explaining this, this increasing trend of lead contamination. But something else does, which is not the behavior of condors, but the behavior of people. And that's hunting. So, especially pig hunting. So these blue lines are basically saying, well, if you account for shifts in how much hunting pressure there is, basically that accounts for this increasing exposure, and especially pig hunting, which I'll show you very briefly. And so we can also see that basically now, for central, if you account for shifts in the amount and type of hunting, which basically generates lead-contaminated carcasses, you can explain why we see this basically seemingly counterintuitive increasing problem. But so pig hunting has gone up dramatically. One reason this is more of a problem in central is just sort of shown here. Here's pig hunting levels in central versus this area of the condor in the southern California. There's just way more hunting in the central area. There's also a problem in analyzing this, which is that there's sport hunting for pigs, and there's what's called depredation, which is basically killing pigs not for sport, but because they're like ruining your fields or your ranch lands. And so there used to be a very loose enforcement of reporting pigs you killed for depredation. Now there's actually, just in the last 5 years, they shifted the rule. You don't have to tell anybody if you're shooting pigs on your ranch, for instance. And so it's really hard to understand exactly how much this depredation has been increasing, but the thought is it's been increasing a lot. One of the reasons for this, which I'm not going to talk about any more than to say this, is basically feral European swine were introduced to some hunting ranches in California and in Texas, and have basically sort of escaped feral pigs, were escaped like pigs from a farm. And now we have basically escaped wild boars also breeding with them, and that's led to this dramatic increase in the densities of feral pigs, this change in the genetics. I'll just show you this one slide to say, if you account for that, you see this pattern, which is that the time period, basically, corresponds to that legislation. Actually, we've been seeing decreased risk accounting for these other factors, especially hunting. So I'm going to talk about one more tiny thing, which is the last thing that we've done, which I think is going, and actually we just got the page proofs for a paper on this 2 days ago, but we are excited about this, is we also analyzed the other thing we do to try to control lead contamination in carcasses, which is talk to people. There's two different things which are done by outreach groups. One is talking to hunters groups, and one is to give them non-lead ammunition boxes. The amount of effort of those two kinds of effort was quantified for about a decade. And you can basically, for this period of time when you know hunting levels, you know the weather, you know all the other things that influence condor, and you know the amount of outreach, you can actually analyze what was the effect of outreach. And this is showing basically that effect for the central and southern. If you look at how many ammo boxes were given away from low to high, and basically how many hunters you contacted at these sort of like outreach events from high to low, you can see that you can drop the estimated predicted blood lead level dramatically from quite high to very, very low. And that effect is basically holds up the same way for the southern area and the central area. You can do the same analysis for basically survival. And this is survival not for a year, but just for about 2 months, which is basically, and that we did that because there's a great deal of seasonality, obviously, with hunting. So you sort of analyze the effects of hunting outreach over short periods of time. But again, you see, if you contact more people and you give away more boxes of ammunition, you can dramatically drive down mortality, drive up survival. And so this is exciting to us because there are many, many outreach activities that are done for endangered species, and almost never can you connect them back to actually something that is biologically meaningful for the animals or plants involved. So summarizing condors, shifting condor behavior sort of critically determines their lead exposure. So basically becoming wilder in particular and using wild food sources basically it puts them in higher danger. pig hunting and basically changes in hunter behavior is also really a big influence on exposure risk. And those have sort of concealed the relative success of basically legislative and outreach activities in trying to at least tamp down what would otherwise be a dramatic increase in lead exposure. So basically those lead bans as legislation have reduced condor lead risk, just not enough to basically sort of counteract these opposing trends. But we can also show that if we continue or ramp up lead outreach activities, that probably does have the potential to actually counterbalance these other sort of prevailing trends, sort of increasing endangerment for the birds. So just wrapping up all of the studies I've talked about, one of the things that I like to tell my grad students, and they'll remember this, because I say it to them all the time, is you should have some sort of useful tool to bring to the table. More people want to help rare species than have sort of unusual tools to bring to the table to help do that. You also have to take, though, the time, even as a professor, to understand the ecology and especially the management context. So a lot of people learn and do sort of generic analyses, sort of simple things to say, well, the population's going up or down with some degree of certainty. That actually isn't usually very helpful. What's helpful is to try to analyze the real specific goals of management or the problems that managers have. And then I think academics, since I'm an academic, I can say this, I guess, I think we play a critical role in conservation by bringing skills which sometimes take a long time to develop, but also independence. Like it's hard for someone who works for the Nature Conservancy to tell the boss that what they're doing really sucks. They have to do something different. But we have a different paycheck, so we can actually give that message and take that message. And something I haven't touched on, and I'm not going to, is I think there's also a real synergy between doing research like this for real conservation goals, and also doing basic research. So I often get more ideas from my applied projects for my unapplied work and vice versa than I would ever have expected. So that's all I'm going to say, except thank you. And I wanted to thank my family, my friends, my dog, my colleagues, my students, and also postdocs.